Welcome, everyone, and thank you for joining us for the second AGRO Lunch and Learn webinar of 2025. This webinar series is made possible through the generous support of Eurofin's Agroscience Services, and we sincerely appreciate their continued sponsorship. I am Solito Sumalong, Regulatory Affairs with Celesta Bio, an executive committee member of the Agro Division of ACS and chair of the Lunch and Learn Webinar Committee, a role I've been honored to hold since 2021. I'll be serving today as the moderator of this webinar. Before introducing our speaker, I'd like to highlight the benefits of becoming an AGRO member. Membership enhances your science, expands your professional network, and supports your career growth. If you're not already a member, you can add AGRO to your ACS membership or join as an affiliate for just $20 a year, regardless of ACS membership status. Simply visit agrodiv.org and click Become a Member. Another important update, the ACS Fall National Meeting is coming up in Washington, D.C. this August. Formal acceptance of the abstracts and the scheduling will be communicated to the presenters in May by the American Chemical Society. But presenters can reach out to their symposium organizers on abstract status now. Otherwise, please reach out to our program chair, Dr. Ming Ma, for other general questions. The Agro Division is also committed to engaging new and existing members, enhancing communication, and building future-ready resources for the global agrochemical community. Kevin, Edmund, Leah, and the Agro team need your help, so visit agrodiv.org to get involved. A quick note on today's webinar format, please use the Q&A tool to submit your questions and comments, which we shall address at the end of the presentation. Also, let us know in the chat where you work and what you do. We'd love to hear from you, and so you can stay engaged with our tremendous audience and community. All participants must act professionally, respectfully, and cooperatively to ensure a safe, inclusive environment. Bullying, threats, discrimination, or harassment of any kind are prohibited, including actions based on race, gender identity, sexual orientation, or other protected characteristics. Sexual harassment, including unwelcome advances, jokes, or offensive behavior, will not be tolerated. Respect personal boundaries at all times. Violations may result in removal from the webinar and other appropriate action. Now, it's my pleasure to introduce our speaker, Maegan Teets. She joined Smithers in 2021 and has expertise in conducting freshwater and marine algal ecotoxicology testing. Maegan previously held a position as lab technician at the Union College Vivarium. She obtained her BS in biology from Union College in Schenectady, New York. So please welcome me and welcoming Maegan to present Exploring the Essential Ecotoxicology Tests for Environmental Risk Assessment. Maegan?
Thank you very much, Solito, for your kind introduction. I'd also like to thank the AGRO Division for the opportunity to present my webinar in their Lunch and Learn series. And thank you everyone for taking the time out of their days to attend. As previously mentioned, my name is Maegan Teets. I'm an environmental toxicologist and study director with Smithers, located in Wareham, Massachusetts. Today, I will be presenting my webinar titled Exploring the Essential Ecotoxicology Tests for Environmental Risk Assessment. First, a brief outline for today's webinar. I will introduce the 3 test types required for basic aquatic hazard assessment and the common guidelines used for these types of tests. After, I will give an overview of the individual tests and some details about what criteria need to be met for a valid or acceptable study for regulators. Aside from the testing, I will also talk about important physical chemical information and how this impacts test designs for the 3 test types. Then we will touch briefly on the methods of analytical support during the in-life portion of testing and how this affects the reporting of endpoints. Finally, once testing has been completed, we will talk about what potential higher tier testing is needed to support a regulatory submission, depending on the results of these 3 test types. A little bit about Smithers, if you haven't heard of us. We are a leading laboratory with decades of aquatic toxicity experience, providing testing to the crop protection, pharmaceutical, and industrial chemical fields. This has given us experience with a wide variety of compounds that require aquatic testing, including those that are considered difficult. For example, those with low water solubility, those that are adsorptive, volatile, unstable, or mixtures. Additionally, through working with these compounds and performing these types of tests, we have developed a large historical control database of studies to help guide and interpret hazard and risk assessment data. We've recently added capacity for the ecotoxicology studies that we will be discussing today with a dramatic and exciting upgrade to our algae lab. Lastly, the Smithers Group as a whole is proud to be celebrating its 100th anniversary this year. A couple definitions that you will hear me mention frequently in today's webinar. First is the acute test, and this is a test design intended to show the effects of a short-term exposure of a test substance to an organism, and this is typically only a few days. Next is the exposure period, and this is a duration of time that the test organism is exposed to the actual test substance. Depending on the test design and organism, this can be from 2 to 4 days. The LC or ECX value, this is an endpoint and it stands for the lethal or effect concentration X. So the concentration at which X% effect is observed compared to the control. The NOEC, this is also an endpoint and stands for the no observed effect concentration. This is the highest concentration in a test at which no significant effect is observed to the control group. Last, and certainly not least, but arguably the most important of these terms, is the functional water solubility of a test substance. This is the water solubility of the substance in the matrix or the dilution water used in the test. And this is not the same as the formal benchtop water solubility value. Before we begin, we have a poll for the audience. You should see the responses pop up on your screen, so please answer in the allotted time. All right, so we have a little bit of a spread. So we have some swinging never, and we have some leaning towards sometimes and regularly. So hopefully those who are swinging towards never, I hope that after today's webinar, you guys feel more comfortable and knowledgeable in this field. And those who answered towards the sometimes and regularly, I hope that this webinar might still be kind of like a review, but I hope that you still take something new and learn something from this time. I'm going to close that and we will move on. So the testing required for basic aquatic hazard assessment, there are 3 tests required for this, and they are microalgae, invertebrate, and vertebrate. They are chosen because they are surrogate species, and they are representative of the local species or ecosystem. They represent the 3 trophic levels of the food chain, so the algal producer, the grazing zooplankton, and then the planktivorous fish. They are selected based on their sensitivity to chemicals, their ease of use in the laboratory, and the standardized test methods that are widely applied and accepted by regulators. This slide lists a few of the common guidelines that we use for the 3 test types. The country in which the product is being submitted for registration in and the end use of the product will affect which guidelines should be followed during in-life testing. The 3 that we test under the most frequently at Smithers are the OECD guideline, which is accepted globally, the OCSPP guideline, which is the United States, and lastly, the JMAFF guideline, which pertains to Japan. The first test that we will look at today is the algae test. So this is a static exposure, and the test duration will vary based on the guideline. So the OECD and JMAFF guideline necessitate a 72-hour test, whereas the OCSPP guideline calls for a 96-hour test. We typically inoculate our algae test at 0 hour with 10,000 cells per mil, since this is a common inoculum allowed for multiple species across all guidelines. This can really change depending on the species used. So other examples for inoculum are 5,000 to 50,000 cells per mil. But again, the species can change. But this will depend on the species, excuse me. The test endpoints that are investigated during this testing are yield, growth rate, and the OCSPP guideline only includes area under the growth curve. The primary goal for these endpoints are the EC10, EC50, and the NOAC. I've included the EC10 and the NOAC here because we have seen that they are of heightened importance to pharmaceutical compounds due to the recent enactment of the 2024 European Medicines Agency guidance. While they may not be as important to industrial, chemical, and crop protection products, they are still investigated during this type of testing. Each of the 3 tests that we will talk about today will have performance validity criteria that need to be met in order for a study to be considered valid to regulators. Control performance validity criteria for algae testing are that we must see a certain cell density increase over the exposure period. For a 72-hour test, we must see a 16-fold increase. At test termination, we typically see a 100-fold increase for our green algae. For a 96-hour test, we must see a 100-fold increase. Again, for our green algae, we typically see a 250 to a 300-fold increase. For a 96-hour test with marine diatoms, we may see a 30-fold cell density increase, but this is OCSPP only, as the OECD does not specify for a marine diatom. The OECD also has some additional criteria regarding the variability of the control data, and that is that the CV, or the coefficient of variation for the section-by-section growth rate, so this is the day-to-day growth rate, must not exceed 35%. The CV for the average specific growth rate, so the overall growth rate, must not exceed 7%. These are the common species used for algae testing. The 4 bolded ones are the ones we currently culture in-house and frequently use the most. 4 species is also needed for US registration for crop protection products, and we will use the 4 species we culture to fulfill this requirement. Most studies in general will use green algae, hence the asterisk. You'll see from the pictures that the green algae and freshwater diatom are unicellular, while the cyanobacteria and marine diatom grow in these nice long chains. We do have methods in place to break up these chains for ease of counting during testing. The next test that we will look at is the invertebrate test, which is the Daphnid acute. This can be a static renewal, or flow-through test design, and this will be flexible based on the test material characteristics. This is a 48-hour acute test, and it is initiated with less than 24-hour-old organisms. This is done by isolating sexually mature adults a day prior and then collecting offspring the next day. The test is typically 4 replicates of 5 organisms per treatment and control. The endpoint measured during this test is immobilization. So immobilization is defined as those organisms not able to swim within 15 seconds after gentle agitation of the test vessel, even if they can still move their antennae. And the EC50 is the primary goal. The acceptability criteria or validity criteria of the Daphnid acute is that the immobilization in the control must not exceed 10%, and the dissolved oxygen at termination in the controls and treatments must be greater than 3 milligram per liter. Other species that can be used for invertebrate testing are listed below with their guidelines. C. riparius, a freshwater insect, is needed for testing for insecticides for the EU, in addition to daphnids. Lastly, D. pulex and C. dubia can be tested under the same guidelines as D. magna. The final test that we will look at is vertebrate or fish testing. Again, this can be static renewal, or a flow-through test design, and the test design will be based on the test material characteristics. This is a 96-hour acute test and typically consists of 1 to 2 replicates of 7 to 10 organisms per control and treatment. The endpoint evaluated is survival or mortality, and the primary goal is the LC50. Please note here that it changes from the effect concentration to lethal concentration. The acceptability criteria for fish testing is the mortality in the control must not exceed 10%. but this will change to 1 fish if less than 10 fish per control and treatment are used. Lastly, dissolved oxygen must be greater than 60% of saturation in the control and treatments throughout the exposure. Common species for this testing are listed below, with the ones we use the most bolded. So those are the rainbow trout and fathead minnow for freshwater species and sheepshead minnow for marine. Depending on the test type and regulatory group, other species of fish may be used as well. You'll see a lot more flexibility with this test design based on the heightened focus on reducing vertebrate usage. I have included the 3 R's here at the bottom and what they stand for. So the first R stands for reduce. An example of this would be using only 1 replicate for each control and treatment, and this replicate would only contain 7 fish. The next R stands for refine, and so we would conduct the algae and daphnid testing first, and then we would conduct a fish limit test at the EC50 of those tests to reduce the overall number of fish used. The last R stands for replace. So if the regulators would allow, we would replace this fish study with in vitro testing, such as the trout gill assay. Also required by some guidelines are the results of reference testing. These are exposures performed with a known reference toxicant that has a large historical database of results. Reference testing is conducted for algae and daphnids, but not done for fish. This is to reduce the amount of vertebrates used. Reference testing is used as a measurement of the health and sensitivity of the organism over time to see if these 2 factors are consistent. It is helpful when introducing a new strain or population brought into the lab to see if the organism behaves as expected based on the historical data. For example, when bringing a new culture lot of algae into the lab, we perform a reference test with it and compare the results to our historical EC50 values. We have reference test data on all our algae species that goes back yearly to 2005. And similarly, we have daphnid reference test data that goes back to 2009. Separately from Smithers, there are published toxicity values that we can also compare against to evaluate our organism performance externally to gain a helpful baseline when we do not have historical data. Lastly, this is also a helpful indicator if a change in practice or procedure has made an impact on the health, sensitivity, and performance of the organism. Now that we have covered the 3 tests, we will begin to move on to the chemical information side of things and how they can impact choices in test design and solution renewal schemes. Having the information listed below is very helpful to have prior to starting in-life testing, since it will help determine if a program will be complex in nature, and it helps us to understand how a material may react under testing conditions. First, and most notably, is water solubility, since aquatic testing should not be designed or conducted above the actual water solubility of the test material. That being said, benchtop water solubility does not equal the functional water solubility limit. Water solubility following OECD 105 is typically performed in distilled or buffered water, which is not the same as our dilution water used in aquatic studies. Other factors, such as the ones below, will drive exposure system selection based on the stability of the test substance. So the compound structure of the test substance, the pKa value, which is a measure of how acidic a substance is. This is calculated from the acid dissociation constant of a solution. The log P value, the adsorption potential of a substance, hydrolysis, the substance's stability in aquatic matrices, photolysis, the substance's stability in light, vapor pressure, which will tell us if a substance is volatile, and finally, if a substance is biodegradable. Before in-life testing can begin, a method of dosing solutions must be established. The method of dosing and the duration of mixing will be based on water solubility and the log P value. Typically, test substances with a lower water solubility and higher log P values will need more energy or time mixing to solubilize. We utilize three methods of solubilizing substances, and those are direct addition, the use of a co-solvent, or the use of a saturator column. Direct addition is typically our preferred method since this is just directly adding the substance to the test matrix. A co-solvent can be used if direct addition is not feasible. Some examples of the solvents we use are dimethylformamide, acetone, and triethylene glycol. The solubility of compounds and solvents is typically minimal up front, but may need a visual trial. And a co-solvent might be avoided and not a first choice in some situations due to the addition of a solvent control to the exposure, the potential to confound functional solubility, or exceed the solubility of the test substance, both which may bring into question the biological results. Additionally, the guidelines will limit the amount of solvent used in a test, which is typically 0.1 milliliters per liter. If using direct addition or a co-solvent, we can solubilize the test substance with the below methods, so high speed or energy mixing, slow stir or extended mixing, and then finally ultrasonication. Sometimes we must utilize a combination of two or more of the above. If we cannot solubilize the substance with these methods, then we would look to use a saturator column. In some cases, a test substance may have characteristics which make it unstable and degrade over the exposure period. In such a case, measures would be put into place to maintain concentrations over the duration of the test. The first two characteristics in the table are in regards to dosing, whereas the remaining characteristics are related to maintaining concentration. So for water-soluble compounds, we would look to directly add them to the test matrix. And poorly soluble or hydrophobic compounds, we would first try to directly add them with high energy mixing or use a co-solvent and a saturator column as we talked about on the previous slide. If a substance is volatile, we would perform a closed system. So the organisms would be in vessels with no headspace versus the typical open vessel. If we have a photodegradable compound, we would perform it in a low laboratory lighting environment, or the testing would be conducted in the dark. The OECD guideline actually allows for the DAFNID acute test to be conducted in the dark for cases like this. For hydrolytically unstable compounds, we would pH adjust the media used. For biodegradable compounds, we would put into place a strict plan for vessel hygiene, so a cleaning process to prevent bacterial growth. For adsorbing compounds, we would condition the glassware used for the exposure to fill the adsorptive capacity of the vessels, or we would look to use alternative materials instead of glass, such as PFTE and HDPE. For multi-component compounds, this would have to be a direct addition to the media or a WAF, and this stands for a water accommodated fraction if poorly soluble. A co-solvent in this case is not recommended for multi-component compounds because it could influence the solubility of various components. For example, one component would be solubilized and the others not. These procedures are really important to put into place since maintaining concentration over the exposure period as much as possible is a stipulation in most guidelines. If stability over the exposure period is unknown, then a solubility stability trial is recommended prior to in-life testing. The functional water solubility can differ from the reported water solubility on an SDS due to a variety of factors, such as the matrix used, the temperature, mixing time, and energy applied. We would first start with a visual benchtop trial to visually estimate the limit of solubility in the dilution water. The goal here is to find the maximum limit, and we would typically start with the guideline maximum of 100 milligram per liter and work down from there until we achieve a visually soluble solution. At this time, we would use the methods we talked about earlier, first direct addition and then potentially co-solvent if needed to solubilize the substance. Once we have a method to solubilize the substance, we would move to the analytical portion, which can be flexible. For example, one high concentration and one 10-fold lower, so 110 milligram per liter. Solutions would then be sampled for analytical measurement at varying time points for evaluation of solubility and stability. and these time points would be decided based on the test that the trial would support. Results from this trial will then help make an informed choice of exposure system design for preliminary and definitive tests. Now that the stability of the test substance is known, the exposure system design can be selected. The first system is a static design. So this is used for compounds that are stable for 48 to 96 hours, and concentrations are expected to remain within 80 to 120% of nominal over the exposure period. This design includes no solution renewals. So once the organisms are introduced to a solution, that is the solution that they stay in for the duration of the test. The next is the static renewal, and this is used for compounds that are stable for 24 to 48 hours. The renewal frequency is designed to generally maintain concentrations at 80 to 120% of nominal, and we would renew halfway through the exposure period. So at 24 hours for a 48-hour test, or at 48 hours for a 96-hour test. Finally, we have the flow-through test design, and this is used for compounds that are stable for less than 24 hours, and this can be intermittent flow or constant flow. We would look to use this exposure system if the decline of exposure concentrations is greater than 20% over a 24-hour period in static or static renewal exposure systems. Please note here, though, that we are not able to do this test design with certain test organisms or certain test substances. After a dosing scheme and the exposure system has been selected, we can now move forward with in-life testing. Dose levels for definitive studies are intended to elicit both a 0 and 100% response, but we are not always able to do this. In order to select appropriate levels, we would begin with a range-finding exposure, which is a smaller scale exposure with a wide dilution range. For example, 10-fold dilutions and often only 2 replicates. Instead of performing a rangefinder with organisms, definitive levels can be based on literature, information on compound toxicity, and mode of action, but this can be risky. Definitive test levels are arranged in a geometric series, usually 32 to 60% dilutions, depending on the guideline, and they generally consist of 5 treatment levels plus the appropriate controls. Using wider dilutions will also increase the confidence limits of the EC values generated. In some cases, the confidence limits may be too wide to define the EC50 value, so keep this in mind when deciding levels. Additional treatment levels can be added if the expected dose response is shallow or a partial response is seen over a wide range of concentrations. A limit test can also be performed using only a control and one high treatment level, usually at the guideline maximum concentration of 100 milligram per liter, or at the test substance's limit of functional water solubility. This can reduce time, cost, and organism use, but comes with risk. If any significant effect is observed, a repeat definitive with complete dose levels must be performed. During the definitive exposure, analytical support will be performed. The analytical method is used to measure the test substance in solution, and if no analytical method in water is available, then one would need to be developed and then validated prior to in-life testing. Analytical support is 1/2 of the total aquatic test, and today there is more rigor and scrutiny applied to the analytical phase of these aquatic tests. It is really important to have a robust and reliable method in place to execute these studies. The Santé guidance allows the conduct of the method validation during the actual definitive study with concurrent control samples, and this is usually our go-to approach as it also covers the United States requirements. The method validation is conducted for the purpose of determining the linear range, detection limit, and the stability of extracts and stocks. During the definitive study, quality control samples will be prepared at each analytical interval. They are prepared at concentrations spanning the range of concentrations used in definitive testing, and also prepared in the same matrix, so the same water, soil, etc. They are used to determine if the method is performing as expected and to confirm the accuracy and precision during testing, and also the repeatability of the method for analysis under testing conditions. After the in-life portion of the study has terminated and the analyses completed, we can determine if we should report our test concentrations as nominal or measured. We can only report concentrations as nominal if the test substance was water-soluble and shown to be stable over the exposure period. Typically, we would report test concentrations as measured, since this is a requirement for GLP studies following all guidelines. The sampling scheme and the stability of the test substance will determine which of the 3 mean measured concentrations listed on the slide we would report as. For algae testing, we typically sample at 0 hour and test termination as a baseline. And if a compound is not stable, additional intervals would be added to better define the decline curve. For Daphne and fish acutes, this gets a little more complex. For static exposures, we also sample at 0 hour and test termination, so 48 or 96 hour. For static renewals, we sample at the beginning and end of the longest renewal interval. For flow-through systems, we sample at the beginning, middle, and end of the exposure as a typical sampling scheme. If the sampling intervals were evenly spaced and the concentrations were stable, so 80 to 120% of nominal, we would report the concentrations as the arithmetic mean. If the sampling intervals were evenly spaced and the concentrations were not stable, so we observed declines, we would report the geomean concentration. If we had an uneven sampling interval scheme, then we would report the concentrations as the time-weighted mean concentrations. Included in the report will be the endpoints which were discussed at the beginning of the webinar. The most common endpoint to be captured was the EC or LC50. This endpoint will be reported along with its corresponding upper and lower 95% confidence intervals. Confidence intervals evaluate the degree of scatter of observed values in the concentration response curve. In addition to the EC or LC values, the NOAC or no observed effect concentration will be reported. Lastly, the slope of the dose response curve will also be included in the reporting of the test results. The slope is the evaluation of the range of sensitivity to the chemical in the test population of organisms. For example, if a flat or shallow slope is observed, this would correspond to a large change in dose causing a small change in effect. This indicates slow adsorption, rapid excretion, or detoxification in the test population or species. This is represented by line A on the graph. Conversely, if a steep slope is observed, this would correspond to a small change in dose causing a large change in effect. This indicates rapid adsorption or rapid onset of the effect. And then this is represented by line C on the graph. Now, I'm not going to spend too much time on this slide since conducting testing under GLP or good laboratory practices is a requirement for regulatory submission, but there can be pros and cons for conducting a non-GLP test. Conducting testing as non-GLP can be beneficial when looking to perform screening studies. Non-GLP studies are often quicker and more cost-effective to perform while still operating with some aspects of GLP testing. Non-GLP testing can also be more flexible and can include analytical sampling. These tests can really be tailored to fulfill specific needs and will oftentimes be used as exploratory tests. Now that the in-life testing and reporting is complete, where do we go from here? And when is additional work potentially needed? Chronic or higher tiered aquatic tests will vary by industry and is driven by length and magnitude of any potential exposure. For pharmaceutical compounds, the algae test will always be a core requirement. The Daphne chronic test and the fish early life stage test will be conducted instead of the Daphne and fish acute. For crop protection compounds, acute and chronic aquatic testing will always be a part of the core risk assessment. And depending on the type of compound, results of the acute and chronic testing, additional testing can be required. For example, the fish full life cycle test and an additional test species for a species sensitivity distribution test or SSD. For industrial chemicals, Chronic testing can be required based on the results of the acute tests. So if high levels of toxicity were observed in the ballpark of an LC or EC50 value of less than 10 ppb, or by production limits. So to wrap things up, the three main basic test types for aquatic hazard assessment are algae, Daphne acute, and fish acute testing. Depending on the results of these tests and the end use of the product, more testing, such as the ones we talked about on the previous slide, may be required. We've briefly talked about the basic overview of each of these test types and what their primary endpoints are. During the definitive portion of the in-life testing for each species, analytical verification is performed to measure the concentrations of the test substance in solution. In order to perform this verification, a method validation must be done prior to in-life testing, and this is done to determine the linear range, detection limit, and the stability of extracts and stocks. After in-life testing is complete, depending on if the concentrations have remained stable over the exposure period, results can be reported as nominal or measured concentrations. Finally, I wanted to share what we as CRO feel is important or helpful information to us to have at the beginning of programs. At the proposal stage, please provide as much information on the compound as possible. This includes, but is not limited to, all available physical chemical information with a special emphasis on water solubility. While we've just learned that this value does not equal the functional water solubility in the testing media, this is a helpful benchmark to have since we cannot exceed this value in testing. Knowing the physical properties of the chemical up front will help establish if a program or testing will be complex in nature and if additional effort will be needed, such as a solubility and stability trial. Also at the proposal stage, please provide if there is an analytical method in water. If not, then one will need to be developed prior to in-life testing. This also allows for establishing accurate timelines for testing and aligning expectations between the CRO and client for execution and completion of the program. Everyone who registered today will be receiving a PDF of the slides via email, and the next slide will also have a list of additional resources.
Yeah, what an interesting presentation. It's certainly generated a bunch of questions that we will get to for the balance of the hour.
Yes, yes. I saw them coming in.
Yeah, don't let that distract you, of course. So we'll close the poll in 5, 4, 3, 2, 1.
All right, perfect. Okay, so
Right.
these are the additional resources I just mentioned. Again, you'll have access to these in the PDF of the slide deck. And I guess we will move on to questions and comments.
Awesome. Thanks so much, Megan, for your presentation today. Like I said, it's great to have this intelligent audience talk about ecotox testing, particularly as it's a requirement for pesticides on a global basis, and it presents, the 300-pound gorilla in the room that a lot of people do talk about, and from a strategic standpoint for those people looking to register technologies into the U.S. and globally, really, for that matter. It's a really good overview. So I did want to group a bunch of questions that we had early on,
Sure.
particularly for species selection, right? We had from Santos, how many fish do Smithers typically use for fish acute range finders? Are there companies who intentionally choose a reduced number of fish, right, for fish acute range finders to reduce the number of vertebrates tested? And I guess I could also have you answer these questions in terms of how these decisions are made within your team, because it's not just you that makes all these decisions. You do have a team involved. So Yeah, I'll let that,
Sure.
I'll leave that to
And
you.
I would like to, preface this with my background is primarily with the algae testing. But as far as I'm aware, so when it comes to selecting the fish for the testing, it will, vary based on the protocol and the country in which the product is being submitted for. So, based on if it's going to be a freshwater or marine species. Regardless of whether the reduced number of fish are going to be used in a test, I think it really boils down to the client's preference. I don't think that it's necessarily going to come from the CRO side of things, as far as I'm aware. But I think it would be an interesting question to pose to some of our experts here as well, some of our fish experts.
Uh, we had a question, and I queued it as answered, but I'll bring it back up. Does Smithers offer testing on macrophytes such as lemna and myriophyllum?
So yes, we do offer testing on lemna. We culture lemna gibba, the aquatic plant. We did not include that in this overall broad speaking presentation, but we do offer that testing.
Right. Okay. Great. So back to the ability to reduce fish species tested. I know you talked about non-GLP studies, right? I know you uh, talked about some of those cost reductions. There was a question about those
Mhm.
costs. But tell me a little bit in general about any opportunities for in vitro testing or any opportunities for artificial intelligence uh, to steer the decisions on testing, as well as the kind of clients or projects and the maturity of the technology
Mhm.
that would steer us into that non-GLP scanning mode.
Sure. As far to my knowledge, I know that we are definitely looking into the use of AI, in our testing. It's definitely, an exciting and innovative kind of like branch, definitely with where the world is going with it. It's going to be interesting to see how it's going to affect this industry, the ecotoxicology industry, and where that will go. I don't know at this point in my position where we are currently in that. Um, Moving away from that, I think that, can you repeat the first part of that question?
Sure. Yeah. I mean, we do have these tools, right?
Mhm.
In vitro
Oh, yeah, yeah.
non-GLP scans that can perhaps reduce time burden for number of species used
Yeah,
in a
so
circumstance.
the in vitro testing can be used to replace the vertebrate testing in this case, should the regulators allow. And that's definitely to reduce the amount of vertebrates used. And I think that's a really good way to fulfill that or to meet that requirement. Not necessarily that's a requirement, but it's definitely something that regulators are looking at. At this moment at our Massachusetts facility, I don't believe that we are doing in vitro at this moment. Granted, again, this is coming from the algae expert, the
Right.
algae person. Um, so yeah, yeah.
Excellent. We can probably bunch a series of questions as it pertain to the exposure system
Sure.
selection, right? I see a question on uh, in the insurance of exposure solutions that don't contain undissolved material, such as an emulsion, right? So you have oil bits in there that probably need to be maintained as at least uniformly dispersible in your test system, right?
Mhm.
You have perhaps substances that absorbs onto some of your machinery or
Mhm.
test vessels. You have you have the idea of closed systems
Mhm.
and volatile compounds as part of your test system. So, uh, uh, walk us a little bit through that in terms of your system selection.
Sure. So undissolved material, especially for the adapted acute and like the fish acute testing, is something that we need to ensure doesn't make its way into the systems because we want to make sure that we are testing the chemical toxicity rather than the physical toxicity of the test substance. So when we're performing all that pre-work prior to in-life testing, that is something that we're looking for to make sure that when we have a solution, everything has been dissolved and is in solution. I know, Salida, you mentioned kind of like those oily substances. In my experience with working with those types of compounds, those kind of fall into that WAF situation, so that water accommodated fraction. How we would typically make those is that we would put the compound in our dilution media and we would mix it for an extended amount of time. So typically it could be as long as a couple days. And then we would siphon out that soluble middle portion because typically with an oily substance, it either sinks to the bottom or it sticks to the top of the solution. So you can see that kind of oil slick. We would siphon out that middle portion where you kind of ensure that there's no undissolved material. And then we would sample that for analytical measurement. And then that's what we would consider our soluble portion of that's what we would use to actually expose the organisms to. So that's kind of how we would address, oily substances. Algae testing can get a little different because the undissolved material doesn't necessarily affect the algae in the same way as it would a fish or a daphnid. It won't clog the algae's gills or, the daphnid's ability to swim. So the algae, same thing. We avoid undissolved material as much as possible. The method we count our algae tests at Smithers, we employ a particle counter for our freshwater species, and then our marine species we count by hand using a hemocytometer and microscope. So if undissolved material is present, we can make note of this because it's not necessarily going to impact the algae as much as it would a daphnid or a fish. If there's undissolved material with our particle counter, we have methods in place to kind of subtract the background noise of this undissolved material from our actual cell counts to make sure that the cell counts that we're generating aren't artificially inflated by this undissolved material.
Yeah, a follow-up to that one, right,
Sure.
Maegan? You mentioned some of the information that you need to really get to hone the test system properly, solubility in water, etc.
Mhm.
In terms of to do all this exploratory work that you're talking about and to run the actual GLP tests, what kind of volumes are you looking for in terms of sample size? Because at the end of the day, you do have potential clients that haven't scaled up too much. That's certainly an issue for a lot of clients in my world.
Sure. So when you talk about solution size, are you talking the solutions for the organisms or the solutions for
Uh,
the analytical
this
sampling?
the actual the samples. Sorry.
The samples, so depending on the analysis, this can be anywhere I've seen from 5 milliliters to 200
Okay.
milliliters. So if we were doing kind of like a direct aqueous kind of analysis, that would be on the closer end of like a 5 milliliters. But if we were doing like a liquid extraction, we would need a larger volume to sample.
Okay. Right. Right. Uh, let's move on to reporting.
Sure.
We do have a question open about under what circumstances will a regulator use time-weighted averages to calculate concentrations? I think we could twist that around to what your approach at Smithers would be to justify time-weighted averages to
Mhm.
calculate concentrations. I think that's more relevant to that issue than
Mhm.
how it was asked.
Sure. So in the situation where we would use a time weighted mean concentration, I'll use the algae example. If we had sampling intervals at 0, 24, and 72 hours of exposure, because we had an uneven sampling scheme, that's kind of a case where we would have to use that. If we had a sampling interval at only 0 and 72, because our sampling intervals are evenly spaced, that's a situation where we could use the arithmetic or geomean, depending on if the concentrations were stable. It will really depend on if our intervals were evenly or unevenly spaced. That will kind of depend on either you're going to use arithmetic and geomean or time-weighted mean. Because the time-weighted mean will take into account the amount of time that has passed in between the sampling intervals.
Okay, right. A very generous comment from Don Walkhope on the questions. A wonderful presentation, incredibly valuable data from Smithers,
Thank
and
you.
also another one that's intending to use this presentation as as a teaching tool. That's fantastic.
Thank you.
So. Under what circumstances does Smithers actually publish? Uh, because
Um,
I do know that there are a lot of agencies on a global basis that can accept publicly published data and uh, and uh, and testing protocols.
um, we definitely have published, I think, in my tenure here, since I've joined the company, I don't think it's happened too often. I think since COVID, it's kind of dropped off a little bit, but it's definitely something that we are looking to get back into doing.
Right.
I presented recently, or 2 years ago now, back in 2023 at CTAC North America. Granted, that wasn't necessarily published, but it was a presentation. We're still kind of active in those roles. So the ecotoxicology side, so presenting at CTAC North America and Europe, and then additionally on the chemistry side of things, so at the ACS meetings and also in like the residue chemistry side of things at those meetings. Um, Publishing, I would say the biggest one that comes to mind would be one of my mentors, Gil or Katharina Sofchek, if you know her. She did a lot of really
Right.
good work regarding the oil spill, so the BP oil spill a couple years back. But that was back in like 2016, I think her work was published. So I think that's the biggest one that comes to mind when it comes to Smithers publishing work.
Okay. Great. Uh, I did want to go back to the GLP versus non-GLP
Sure.
question just a little bit. I know there's a cost difference, and there's probably a multiplier involved. In terms of time, how
Mhm.
much do you save in that under that circumstance? I guess that would be the other part of that question.
So for time, so for non-GOP, you can choose to perform a rangefinder and a definitive, or some clients will prefer just to perform a definitive study. So that kind of takes away that rangefinder. On the reporting aspect side of things, the reports will tend to be really pared down. So you won't get that full kind of report that a GLP study would get. Normally, we would just prepare a summary report. And those will typically take much, much less time than a normal report would take to issue to the client.
Right. So we have herbicides definitely in play when it comes to the world of pesticides. It would seem like most herbicides would kill algae, and you're in the algae world.
Mhm.
How's that play into your design and registration of these tests?
So yeah, you're correct in that, the herbicides definitely kill our algae. It's definitely with those compounds that you'll see us testing very low. So we're talking probably microgram per liter. Um, that's definitely something that we have seen a lot more. We've seen a lot more toxic compounds coming through recently. So it's definitely kind of made us put our heads together to figure out how we're going to test them. I don't think I mentioned it in the presentation, but the use of solvent can be really helpful when we are testing low because, trying to make a solution at, 10 ppb is kind of difficult through direct addition. So then solvent can be helpful because the solvent stocks are actually much, much higher. So we're in the units of like milligram per milliliter rather than per liter. So that they can be helpful in dosing solutions at those levels.
Okay,
So the
great.
herbicides usually are much, much lower for algae testing, whereas they might not be as low for the fish and daphnids.
All right. And yes, I wiped a question I didn't mean to, but are these OECD guidelines and probably the OPPTS guidelines applied for testing microbial pesticides as well? Are they the same or are they different between conventional chemistry and
Um,
biologicals?
I would have to get back to you on that. And that might have to be one that I would have to check with our
Okay,
experts on.
right. Yeah, I know. I know in the
Mhm.
U.S. it might there might be some variance. I would have to look that one up myself. So,
Yeah, yeah.
all right. Well, thank you so much. Thank you so much, Megan, for your kind presentation. Let me
Thank you.
share my share my screen just real quick. Here we are. Yeah, a special thank you to you, Megan, for your excellent presentation, to Eurofins Agri-Science Services for their generous sponsorship, and to Kyle Sawyer, our dedicated webinar coordinator. For any questions we didn't get to, we'll be posting responses in the coming days, so stay tuned for those updates. You may also receive a follow-up survey, and we'd greatly appreciate your feedback to help us continue improving this series. Mark your calendars. Our next webinar is on May 7, featuring Prashesh Sharma from BASF, who will present a panel on sailing in the waters of FIFRA data compensation, another 300-pound gorilla in the regulatory world in the US. We'll also have additional webinars lined up on Wednesdays at noon in September, October, and November. If you have ideas for future webinar topics, we'd love to hear from you. So reach out to me, the chair of the webinar committee. The Agro webinar series is a fantastic way to introduce new audiences to Agro's incredible community and showcase the groundbreaking science within our division. It's also a great opportunity for anyone interested in learning about new areas and even stepping in as a future moderator. So thank you again for your participation, and we'll see you again on May 7th. Have a great week, everybody.